Hello? So I don't know why it decided not to work today. So we just have to do this manually. So yeah, no, it's not working. Okay, so how was the break? Did you take advantage of your time off to do fun stuff? Like studying in other courses? Okay, so last time we stopped in the part where we explained about the transcription process. About the transcription, I mean the conversion from DNA to RNA. And now we'll talk a little bit about the structures that RNA can take. As you probably saw, that RNA is single-stranded, or mRNA is always coming in the form of a single strand because it's being copied or transcribed on the basis of one of the strands, and it's a copy of that strand. And because of the single strand structure, it can form these structures we call secondary structures that are because of the interaction between the bases of this mRNA molecule. So here it's not depicted, but you can imagine that all along here you have these bases that can form, like in DNA, they can form hydrogen bonds between them and form these hairpin structures, which are very common, as well as stem loop structures. And there are also more complex structures that arise from three-dimensional folding. And one of the examples of these structures in a molecule that is called tRNA, or transfer RNA. This is what we can see here. And this molecule mediates between, it can know how to couple between a specific amino acid that's normally bound to this region over here, and a codon or triplet of mRNA that knows how to read on the other side, thus translating the genetic code to a sequence of a protein. So the roles of RNA in general in the cell, we can summarize it as that mRNA, general messenger RNA, is involved in information processing and transfer. Then you have the tRNA, that is the molecule that does the sequence interpretation. And here you can see a different example. This is the tRNA molecule. It's a very simplified model of the tRNA. And in one end, it has what we call an anticodon that can recognize a specific location on the mRNA. And on the other side of the molecule, it has an amino acid that it later knows how. And inside this whole structure that's called the ribosome, there's a peptide bond that is formed between the amino acid chain and a new amino acid that comes along, and then the peptide grows forward and is guided by the sequence of the mRNA as at each codon. You learned about codons, right? Like a little bit in the previous classes. You learned a little bit about the codons, right? So this ribosome, like we also discussed in previous classes, is made of mostly RNA, not only RNA, but mostly RNA, and the RNA that is formed by the ribosome that forms the ribosome is termed rRNA, or ribosomal RNA. And there's another population of RNA that we'll discuss in further classes, which involve in regulation of expression. RNA that I told you before are termed non-coding and are not being translated to proteins. And you have short and long non-coding RNAs. So these RNAs have catalytic activities and they can regulate expression of other mRNAs. Okay, it's not working. Why am I trying? So, I saw before the genetic code. This is the table that depicts this code. A fact that's very special and amazing about this code is that almost, with the exception of very few aberrations or differences, all living organisms share the same code. Meaning that if in any amoeba or plant or mammal, you will find the translation. If you find in the mRNA the sequence is GUU, you will get valine. Okay, and this is very well kept, and this is one of the reasons that people also support evolutionary theory that everything started from the same father or organism, or some kind of father prey organism. And one of the things that you can notice here is that it's enough in many of the amino acids, the two first letters, in this example, if you want to understand this table, then C is the first letter, U is the second letter, and then the third letter, because biologists don't like three-dimensional matrices. So all the fourth letter in here, as you can see, but here it's actually for a good reason, because one of the things that you can see is that all the codons that terminate with the third letter, it doesn't matter which third letter it is, all four possibilities of the third letter translate to leucine. And this is actually due to it's a mechanism of redundancy, okay? And this is because of specific biochemical properties of the tRNA, the transfer RNA that we talked about before. And this is actually like a proofreading mechanism that makes sure that there is less mix-up between the different amino acids, and also it enables to overcome certain mutations and aberrations that may happen in the genetic material. Why am I trying to? Okay. So, what is translation? In translation, as you can see, like here in this nice GIF, you have the mRNA sequence bound to the ribosome, which is it has a small unit and a large unit, and one by one the tRNAs are coming together, enter through a specific entrance site. Then the peptide bond is formed between the polypeptide chain and the new amino acid that is coupled according to the triplets of the codon, and then this is actually some kind of chaperone. This actually shows like a mechanism of how you can translate proteins also in different areas of the cell. But in general, this illustration shows you how this process occurs. And more specifically, I know it's hard to ignore this for a moment, but look over here, that the ribosome, the eukaryote, or I didn't talk about the difference between eukaryote and prokaryote, but never mind. Our ribosome has three locations for the tRNA to be in. First is the A, where the tRNA first enters. Then you have P, where the peptide bond is actually formed, and E, where the tRNA comes out after it loaded the amino acid to the growing peptide chain. And you have, and this is the general process of the formation of a peptide bond. Okay, so first of all, you have like a specific type of amino acid that always starts. It's always methionine, that identifies the first part of the mRNA, which is called the start codon. Okay, and then you have other tRNAs that come in and bind here. Then the tRNA is transferring the second actually peptide, the second amino acid to the first amino acid, and the first amino acid that was here is moving to the exit site and eventually gets released, and this process is obviously repeated hundreds and thousands of times. So, wait. We'll see a movie to summarize this.
The job of this mRNA is to carry the gene's message from the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for. There can be several million ribosomes in a typical eukaryotic cell. These complex catalytic machines use the mRNA copy of the genetic information to assemble amino acid building blocks into the three-dimensional proteins that are essential for life. Let's see how it works. The ribosome is composed of one large and one small subunit that assemble around the messenger RNA, which then passes through the ribosome like a computer tape.
You can see how old this video is.
The amino acid building blocks, that's the small glowing red molecules, are carried into the ribosome attached to specific transfer RNAs. That's the larger green molecules, also referred to as tRNA. The small subunit of the ribosome positions the mRNA so that it can be read in groups of 3 letters known as a codon. Each codon on the mRNA matches a corresponding anticodon on the base of a transfer RNA molecule. The larger subunit of the ribosome removes each amino acid and joins it onto the growing protein chain. As the mRNA is ratcheted through the ribosome, the mRNA sequence is translated into an amino acid sequence. There are 3 locations inside the ribosome, designated the A site, the P site, and the E site. The addition of each amino acid is a 3-step cycle. First, the tRNA enters the ribosome at the A site and is tested for a codon-anticodon match with the mRNA. Next, provided there is a correct match, the tRNA is shifted to the P site and the amino acid it carries is added to the end of the amino acid chain. The mRNA is also ratcheted on 3 nucleotides or 1 codon. Thirdly, the spent tRNA is moved to the E site and then ejected from the ribosome to be recycled. As the protein synthesis proceeds, the finished chain emerges from the ribosome. It folds up into a precise shape determined by the exact order of amino acids. Thus, the central dogma explains how the 4-letter DNA code is, quite literally, turned into flesh and blood.
So what is missing in this, in the final segment of this video? No? Something that we learned about before? What helps the protein fold? Chaperones. Exactly. Then this is actually obviously simplified, very much simplified this video, but one of the first things, almost a lot of the majority of proteins use also chaperones in order to fold correctly, especially if they're long proteins. So just to summarize this chapter before we move to the next chapter. Wait, it's here. Then show another movie. Today you have a lot of movies. Yeah. Well, it's a good question, because what I like also, I told you about these videos in general, that you saw that there was a bunch of tRNAs there. There's a very high concentration of tRNA. Actually, the ribosomal RNA and the tRNA form like a significant amount of the cell volume, of the cell weight even, in general. So the area where the translation normally occurs is packed with tRNAs, with a lot of tRNAs. And you need to understand that in any biological, when we're talking about biological reaction or a chemical reaction in general, everything is in a state of like equilibrium. So the tRNAs bind just one after the other, and if there's no complementation between a specific tRNA and the mRNA, it just gets released. And just when you have like one of the tRNAs that binds and actually stays stable long enough, then it gets to go to the next stage and the next amino acid is incorporated. Okay, so a lot of time we think it's like a mechanical. You look at it mechanically, but actually there's a lot of statistics involved here in general, and a lot of thermodynamics also. So just to summarize everything,
the DNA double helix contains two linear sequences of the letters A, C, G, and T, which carry coded instructions. Transcription of DNA begins with a bundle of factors assembling at the start of a gene to read off the information that will be needed to make a protein. The blue molecule is unzipping the double helix and copying one of the two strands. The yellow chain snaking out at the top is a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA letter by letter to copy the gene.
So like you said, again, with the nucleotides that need to bind, it's the same thing. Okay, just the nucleotides that are complementary or stay there long enough in order to be incorporated in the growing chain. But there can also be mistakes. Okay, it's not completely.
The RNA needs to be edited before it can be translated into a protein. This editing process is called splicing, which involves removing the green non-coding regions called introns, leaving only the yellow protein-coding exons.
So we didn't get into that this procedure too much, but mRNA is not normally nothing that we teach you here is that simple. You know, everything is more complicated. But in general, mRNA, after it's being transcribed from DNA, it needs to overgo a series of processing, and part of this, one of the procedures or one of the steps of processing that mRNA goes through is alternative splicing, meaning that chunks of the mRNA are being cut out, and this is one of the mechanisms that determines the variation between different genes and different tissues, because you can have a tissue where you have a specific where this chunk may not be taken out, and you have a different type of tissue where this sequence can be cut out, and thus you can get from the same gene many different proteins because they're edited differently in different tissues and different types of cells. Okay?
Splicing begins with assembly of factors at the intron-exon borders, which act as beacons to guide small proteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released, and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes remove all introns so that the edited RNA contains only exons, which are the complete instructions for the protein. Again, this is happening in real time. When the RNA copy is complete, it snakes out into the outer part of the cell.
So this splicing is actually one of the things that is very, humans have the largest amount or one of the, there's a huge difference. A lot of time you hear about that the genome is very similar between us and, amoeba or something like that. And then actually a lot of organisms or higher organisms differ in the amount of splicing, amount of variation that every gene undergoes. Meaning that you don't get from just a single gene a single protein. From a single gene you can get multiple proteins. Okay, so 95% of all the genes in humans are being alternatively spliced. So not the same as they come out from the DNA.
Then all the components of a molecular factory called a ribosome lock together around the RNA. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. Inside the ribosome, the RNA is pulled through like a tape. There are different transfer molecules for each of the 20 amino acids shown as small red tips. The code for each amino acid is read off the RNA 3 letters at a time and matched to 3 corresponding letters on the transfer molecules. The amino acid is added to the growing protein chain, and after a few seconds the protein starts to emerge from the ribosome. Ribosomes can make many proteins. It just depends what genetic message you feed into the RNA.
Okay. So, with this we finish this chapter, and now we'll start moving ahead to biomembranes and cell architecture, which bring us one step closer to start talking about actual like things that are unique in neurobiology, like I know you're all waiting for. And I don't know which one it is. So, actually, specifically in neurons, this when we talk about biomembrane and cell architecture, this chapter will mainly will discuss the bilayer or the cell membrane, which is the border or the defining outline of the cell and what separates the cell from its environment. So, in general, eukaryotic cell, and when I say eukaryotic cell, I mean cells that are not bacterial in general, or like all evolved organisms and all plants and all what you would think about that are not bacteria are eukaryote. So inside the cells, they're divided into organelles. And each organelle is defined as an organelle because it's surrounded by a biomembrane that encloses it. And when we say, this is just for general terms, when we will say a lumen, we will mean the aqueous interior space of an organelle or a cell. In general, you'll hear like we'll use a lot of terms in order to describe the intracellular domain, or cytosol, or many terms that people use to describe the interior of the cell or organelle. And the cytoskeleton in general, which we'll discuss in the end of this chapter, is a dense network of three classes of protein filaments that give mechanical support for the cell in general. In a bacteria, it's just one large space, okay, for everything, and all the processes are more coupled. Like you can have like a mRNA that's being formed, and right after it's being formed, you have ribosomes that binding to it. You don't have the separation between the different components. So everything is just together. And also because one of the reasons that the bacterial cells are much smaller than eukaryote cells, they're like 1 micron, and eukaryote cells are like 20 to 30 microns large. So one of the reasons that you have these organelles, okay, we never say reasons in biology, but one of the logics to have these organelles in eukaryote, because the diffusion of molecules is a relatively slow process. So if a molecule needs to travel large distances, then you might as well capture all the molecules that you need inside a specific organelle. If you want to produce energy or degrade proteins or everything, then you want all the molecules that are relevant to be inside a specific organelle that has a function. You don't want it to be drifting around the cell. But if the cell is very small and you just pack everything in there together, then the organelles don't give a very large advantage. Also, organelles add complexity, and a lot of times simplicity, especially for bacteria, is a very high advantage. Also in bacteria, you have the DNA, the material is not entirely spread around the cell, but it's concentrated to a certain region, but it's not embedded by an organelle. Okay. So, what we'll talk about in this chapter, we'll first talk a little bit about biomembranes, what are they made of, and the structural organization. Then we'll talk specifically about the organelles, the cytoskeleton, and how do you purify parts of the cells and analyze them. So, as I also showed you before, this is an EM or electron microscope image of a slice of a cell, and here you can see a very nice depiction of this. All the what do we call a biomembrane in general, or in eukaryotic cells, is formed from a bilipid or phospholipid bilayer. This is how it's termed. It's because it's formed from two layers of these sheet-like that form this sheet. And these layers are formed from multiple units of these small molecules that are arranged opposed to each other. Each of these molecules has a hydrophobic or a hydrophilic or polar head, and then a hydrophobic long tail, and they form together in this formation. One end is facing the exterior, and one end is facing the cytosol of the cell. And could you maybe imagine, we talked about a little bit in the beginning of the classes about the forces that we have in chemistry or organic chemistry in general. Can you maybe guess what is the force that is holding the structure together? What type of force is holding the structure? Yeah? Van der Waals is one of them, but there is one more important than that. Hydrophobic interactions, or the hydrophobic effect. You remember that we talked about the fact that if you have hydrophobic molecules, they will tend to aggregate to one another because the interactions that because water cages that are formed around them are more stable. If you have less surface area, then this is the main. So hydrophobic effect and Van der Waals interaction between the fatty acyls, which acyl is like a long chain of carbon and hydrogen, like we'll see in a minute. And because of these interactions, the hydrophobic area is compact and stay together, and the hydrophilic is pointing towards the water, because here and here you have a lot of water, and here you don't have any water at all. So I talked about that. And in general, one of the things that this barrier enables the cell is to be very specific about the type of molecules that can enter. Okay, for example, I'll show you later a slide that talks specifically about the permeability. But in general, apart from this simple structure that we see here, there are also very unique structures, especially in regard to the nervous system, that membranes can form. For example, a synapse. Again, these are all EM microscopy. So this is a very nice image of how a synapse looks like in electron microscopy. One of the things that I want you to look at, this is a tissue, okay? And what do you see here around all these regions here? It looks like a mess, right? You have a lot of things, a lot of stuff. A lot of time when we show you in the caricatures, like in these animations and all these things, and you need to imagine that the biological matrix or the biological area of the cell is filled with a lot of proteins and a lot of other cells, so the cell is not just floating around in space. Okay, and this is one of the things that you can see very nicely here. But here they highlighted these two areas, and what you can see here, if you're very and you have this illustration as a guideline, is that you can see here the presynaptic terminal, okay, which is also it's a little bit hard to see, but you can see there's also a bilayer here, right? You see these two lines, and also here you can see in certain regions that you have these two parallel lines. So this is part of the membrane of the cell, and this area belongs to the axon of one cell, and this area belongs to the dendrite of the postsynaptic cell, as we would recall, as we would term it. And here you can also see very nice examples of these vesicles. Did you learn about synaptic transmission in general? A little bit? No? So, in a typical axon, you have in the presynaptic terminal, you have these microvesicles that carry the neurotransmitter, and they're the ones. And when you have an action potential that's being fired, then these vesicles, much of them are fused to this membrane. They're actually made of membrane themselves, and they fuse to this membrane and release the neurotransmitter, which then crosses this barrier here. It's called the synaptic cleft, which is a very tight gap between these two cells, and then the neurotransmitter is received in the postsynaptic cell. Okay, so this is one of the structures that you normally find in a neuron. And another structure that relates to the central nervous system, or the periphery also, is the one of myelin sheath. Did you ever hear about the myelin, like how it's formed and stuff like that? I just want to know, like, in general. Okay, so many axons of neurons in the central nervous system and the peripheral nervous system are what we call myelinated. Okay, and this myelin in the central nervous system is actually caused by the wrapping around of a membrane of another cell, which in this case is the oligodendrocyte, that wraps multiple times around the axon of a different cell. And what it actually causes is an isolation of this area that is being myelinated from the extracellular area. And this is what actually facilitates and enables the transmission of active action potentials to be propagated rapidly and efficiently along the axon, or electrical signals. So here you can see there are multiple layers, and these are all also lipid bilayers. Questions? Yeah.
How exactly the speed is enhanced? I just know it seems like jumps quicker, but why does it jump quicker?
What is it? Well, it's actually what it prevents is from the ions that move along this axis. It prevents leakage, and thus the current that we didn't talk about. We're not talking about this now, but we'll talk about it in later classes about the special mechanism of propagation of action potentials. But in general, if you have in every node, you have the generation of actually a new generation of the action potential. And what you need to achieve is that the current that starts here will not leak outside of the membrane, and so that it will not decay until it gets to the next node and then be propagated once again. So what it actually prevents is it doesn't actually like speed the process, but it allows a fast process to be transmitted without being lost on the way. Okay, but we'll talk about it more specifically when we talk about propagation of action potentials. This is just to show you like a general illustration of how this structure looks like, and you can see that also in real life it's very well organized. Why isn't? No. Okay. So when we talk about organelles in general, we need to also define what we term the exoplasmic or extracellular face of the membrane, and what is the cytosolic face of the membrane, because the two are not equal to one another. And this is an illustration of the main organelles that we have in the cell. And in general, the nucleus is surrounded by two membranes. One internal membrane is actually what defines the shape of the nucleus, and the external membrane also defines the shape, but also spreads out and forms the formation of what we call the endoplasmic reticulum, which is a formation that is very important for proteins that need to be shipped outside the cell, and also generation of lipids, and that will later form and join the membrane. Another structure that we have is the very important structure or organ in the cell is the mitochondrion, which is surrounded again by two layers. It has an internal membrane and external membrane, and this unique structure is what enables it. I don't think we're going to discuss in detail how it does it, but in general, this structure, this is like the power source or the power plant of the cell, and this is what converts glucose or carbohydrates to the form of ATP and energy. And apart from that, you have also a lot of vesicle type organelles as lysosomes. You remember that we talked about, you asked me about how the lysosomes, or you asked me how the lysosomes that degrade the proteins, right? We talked about the lysosomes degrade proteins from the extracellular matrix, right? Proteins that are outside the cell. So this is an explanation of how the lysosome exactly degrades proteins that are outside the cell. You can imagine that the lysosome is first formed by this invagination or entrance of membrane inside. Then this membrane closes, and certain proteins and pumps move, create a very, very favorable environment here for degradation of proteins. And you can see here the black. If the black part is the exoplasmatic phase, then inside the lysosome you will have proteins that were located in the exoplasmatic phase of the cell. Okay, it's inside the cell, but the area here is equivalent to the extracellular domain. Another organelle that we're going to talk about is the Golgi apparatus. Again, that is important as the endoplasmic reticulum, but is more important for secreted proteins and proteins that the cell secretes outside and ships outside the cell. And that's about it. So, as I said, the biomembers are assembled. We can separate them to three types of lipids, or in general, three types of molecules. You have the phosphoglycerides, you have the sphingolipids, and the steroids. All of these molecules are what we call amphipathic. And do you remember what we said about amphipathic, although it's written here? Amphipathic is a molecule that is polar in one side, and or hydrophilic in one side, and hydrophobic on the other side. And as you said before, this is very important for a molecule to have this property in order for it to be embedded in this kind of formation of a membrane. So Obviously, these molecules differ in their chemical structure and their function in the membrane. So, the first and most common type of molecule is the phosphoglyceride molecules, which are basically built from two long hydrophobic carbon-hydrogen tails, which are linked to a glycerol molecule and a phosphate, and they're esterified, which means a bond through oxygen to various head groups. So you can have all these different head groups that are bound to this basic structure, and this is like 80% of all the composition of the membrane is made from molecules that look like this. A second type of molecule that is normally found in membranes are normally also in myelin. In the myelin cover, you have these molecules are abundant. They're called sphingolipids, and the basic structure is this molecule here, and it can have again variations in the head group, and all of these are polar head groups that can be attached to this basic structure here. And the third group of what we call lipids is the cholesterol, and we all know that you all think that cholesterol is bad, but it's actually not bad, and I'll explain why. But having too much of cholesterol is bad, but in general cholesterol is very important in order to keep certain properties of the membrane. And here it doesn't look like the molecules that we have before, but again this molecule is still amphipathic, and it has a hydroxyl group here, which is equivalent to the polar head group in the other lipids, and you have these ring formations, or these four ring formations, which are hydrophobic. So in general, we need to think about the membrane. It looks like it's hard to imagine it from the photos, but in general, the membrane is very fluid. Okay, you need to think about it as like a fluid substance that always changes and always moves. And this fluidity is a property that is very important for the membrane to function. Okay, and also a membrane, like the cell needs to keep a constant pH and temperature and everything, also the membrane needs to be in a specific fluidity in order to function. So most lipids, most lipids and many proteins that are inside the membrane, they are free to move laterally, meaning to diffuse along the axis of inside this membrane. And the viscosity of the membrane is actually like a stupid comment because you can't appreciate it, but the viscosity of the bilayer is 100 times as great as water, meaning that it's a lot more viscous than water, and you can compare it to the viscosity of an olive oil. Okay, in general, so it can give you a feeling that it's not the membrane is not like a solid, but it's actually more liquidy in sense. And you have some compartments in the membrane that what we would term as lipid rafts, just large areas of proteins that are like floating around in the area of the membrane. And obviously the lipid composition influences the physical properties of the membrane, meaning if you have more cholesterol, less cholesterol, if you have more of one type of lipid and more of a second type of lipid, you'll have different physical properties of the membranes. So in general, a biological membrane looks somewhat like this. It has proteins. Some of these proteins are anchored to the cytoskeleton, which we'll discuss later. And you have also cholesterol molecules that are embedded inside, and also not all the molecules that are obviously in the membrane are shown here. So just a short movie about...
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it.
So this is actually what I talked about when I say the hydrophobic effect in general. Like it's a self-assembling structure in general. That's very important. This huge structure, if it would require like enzymes that will build it or stuff like that, it will be impossible. Okay, because it like encases the whole cell. So it's very important for a structure like that to be self-assembling.
Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Okay, so this is just to get the idea of general, like the fluidity of this whole structure, which is again very, very important. So in general, what determines this fluidity? So if you have long and saturated fatty acids, or do you remember that we talked about saturated and non-saturated fatty acids? The ones that actually have an. So this is an example of a saturated fatty acid, and saturated means that all the carbons have hydrogens attached to them, like 2 of them are attached to the neighboring carbons, and 2 of them have hydrogens. And unsaturated are where the situation where the carbon, not all carbons have hydrogens in all the possible connections, and thus forms this kink. Okay, so this is the cis configuration, and we also talked about trans fats. So in general, if this molecule is long and saturated, meaning it's linear, then it will tend to be it will have a greater tendency to aggregate and to be in like a gel-like consistency. Okay. But if you apply heat to this structure, because of the energy in this structure, then you will have a fluid-like consistency. In a sense, it's like you were taking butter and melting it on a pan. Then you're breaking all the interactions between the fats, and you're creating like a fluid-like consistency. So in this example, you have. This is palmatic acid, which is palm oil in general, that in room temperature will be solid, and this is oleic acid, that is olive oil, that in room temperature will be a liquid, and they're actually the same length. So obviously lipid composition affects the thickness and also the curvature of the bilayer. And how is this possible? Is that certain lipids tend to form this steric or formation of like a cone-like shape. This is phosphatidyl ethanolamine, and phosphatidyl choline tends to be more linear or more cylindrical in shape. So many times, if you will find these curvatures in the membrane, they will be formed because you have more of these types of lipids that are around here, and these types of lipids which are in the outside areas of the cell. And also, as I said before, cholesterol can have an effect on the phospholipids themselves. For example, phosphatidylcholine broadens and becomes thicker when it's associated to cholesterol, but types of sphingomyelin do not have this property. But in general, it's important to understand that cholesterol has this buffering effect on the membrane, meaning that imagine that you have a membrane that is too fluid, and now you want to make it more rigid. So if you add cholesterols here to this state, then the cholesterol will form these interactions that will stable this structure. But let's say you have a membrane that is too rigid and you want to make it like more fluid, then if you add cholesterol to this organization, then you can imagine that the cholesterol will actually break a little bit of the associations between the lipids themselves, and thus create a more fluid-like consistency. So the cholesterol is very important in order to control and regulate the fluidity of the membrane. We actually need to take a break. Okay, so take a 10-15 minute break and continue with that next class. כן. כן, הם צריכים להיות מוקלטים. אני לא יודע מתי הם יעלו לאינטרנט, אני צריך לשאול את מי. מה המצב? זה מחר, זה לא עובד. בעיקרון הוא קלט את זה, אבל... כן, החלפתי את זה, יש פה שניים. מה את רצי? מה זה רעיון הפרויקט?
זה פרויקט שאנחנו פתחת אותו.
כבר יכול מוקדם, כי יש להם שירות.
לא, כל אחד חד לדבר 10 דקות. נראה לי כל אחד לדבר את זה. אני הרבה פחות.
אני עדיין לא 100%.
אפשר להיות בסמינר?
לא.
למה? קבעתי תור, מה? אני יושב על ידה, היא תעתיק אותי. כן, תעתיקי ממני? וואי, וואי. מה נשמע?
מעניין. כן, זה לא, זה עבד כל השנה, זה עד עכשיו עבד, עכשיו זה משום מה לא עובד. כי אני פותח מצגת וזה לא... זה לא זה, נראה לי הוא לא חושב שהוא קולט אותו. כי הוא עושה את הרעש, אבל הוא לא...
כמה זמן יש לנו?
5 דקות.